Good morning. Thank you all for coming to hear my talk on my research. Um, I've always been fascinated by animals, and especially insects, since I was just a little girl. Insects are the most diverse group of animals on Earth, and they've evolved incredibly rich repertoires of behavior, a few of which are shown here, and many of which are studied by lab groups here at the zoology department. So you're probably familiar with most of those. You know, they walk, they jump, sometimes they swim, they fly. They can also navigate. They may fight over potential mates. They also woo with courtship songs and visual displays, and some of them even take care of their offspring. And the nervous systems that control these behaviors are complex, but they're also incredibly small, which means that we have the ability to describe and map them even down to the single cell level. So how small are we talking about? Well, if we look at an example, this is Drosphila melanogaster, also known as the fruit fly or the vinegar fly. This whole nervous system, which is the brain plus the nerve cord, which is like our spinal cord, is less than 1 millimeter wide. And if you look at the brain, it's about 120,000 cells total. And if you look at the ventral nerve cord, it's about 20,000 cells. So they're very, very small. Why am I picking Drosphila melanogaster? So it's not as charismatic as butterflies or even bumblebees, but it's actually been used as an experimental organism since the early 1900s. It's easy and inexpensive to raise in the lab. You probably know that they reproduce very prolifically and quickly if you've ever had similar insects in your kitchen. They also have a small number of chromosomes, which was very important for early work when they were used to map genes onto specific locations on the chromosomes. And over a century of experimentation has produced a lot of very sophisticated genetic tools. Why is this important? Because scientists can use them to do all kinds of things. We can visualize specific cells of interest. We can turn specific genes on and off in these cells. We can activate or silence specific neurons and then look at the effects on behavior, and it kind of goes on and on. So I'll just focus on a few things. So like many animals, flies use their sense of smell in order to locate food, mates, resources for their offspring, things like that. And as a graduate student, I began working on the primary olfactory or smell center, which is called the antennal lobe. And if I can point, these are the round structures here on the front of the fly's brain. So we have sensory neurons located at the antennae that get activated by odors from things like a ripe banana, and they will transmit their signals to specific compartments in this antennal lobe. And from there, they'll form connections with partner neurons that are going to carry those signals up to higher brain centers. And these are used in order to produce instinctive responses to odors, and even for learning and memory, for associating odors with specific experiences. So when I was working on this as a graduate student, I really enjoyed studying the genes and hormones that were required in order to organize these neurons during development. But we didn't really know very much about what happened further on in the brain. So, what are the neurons that they're connecting to in order to produce these instinctive behaviors or in order to produce learning and memory? It was really kind of a black box at that time. And many scientists were working on methods to try to capture those cells that they were connected to. But these methods were somewhat limited. So they would sort of capture random occasional cells, but not all of the partners that these neurons had. So what we really needed was a way to find all of the partners and see how strongly they were connected and form some kind of a wiring diagram to understand how smell and learning and memory and so forth worked. And what we needed was a connectome. So what's a connectome? It's a comprehensive map of neurons and their connections that form a network, right? And this can be from either a partial or an entire nervous system. And if you look at mammals, you can only really do at this time a very small chunk of tissue from a brain or a spinal cord and look at the cells in that chunk. But insects are so small that we can actually map an entire nervous system. So we can do this, and then by analyzing this connectome, we can identify potential circuits that are carrying information processing for sensory inputs and for motor outputs or behavior. All right, so how do we construct a connectome? So this is one way. So to build a connectome, we would first preserve and dissect the tissue of, for example, this insect brain shown here. Then we can basically slice it into thousands of thin sections and image those with an electron microscope, and then align all the images into a 3D volume. And we then extract the neurons from these black and white images, and we can even visualize the connections that neurons make with each other. And currently we use AI, so it's an automated segmentation process that basically finds those outlines and then fills the whole neuron. But it's not entirely perfect, so after that we do a lot of proofreading in order to correct any errors from the segmentation, and we also identify the cells, label them, and then start to analyze the circuits. So I'll just show you a movie. So we're basically zooming through an electron microscopy data set with the automated segmentation of each neuron in a different color. And what we're looking at here is essentially the insect compass that's used for navigation through space. Okay, so it wouldn't be enough just to provide those segmented neurons. It's also really important that we annotate them, so annotate different features of the neurons, and also assign them names so that scientists can compare them and can identify and retrieve the cells that they're most interested in. So you can think of this process as being like labeling businesses and streets in Google Maps rather than just giving you the raw satellite data. And this is actually one of the primary kind of goals of the Drosphila Connectomics group here in Cambridge. All right, so I moved to Cambridge in 2017 to work with this Drosphila Connectomics group. It was led by Greg Jeffress, who was actually my lab mate when I was a graduate student back at Stanford many years ago. And my first project here was to look at, again, the olfactory system that I mentioned earlier. So here we have the antennal lobes that I showed you before. What I did first was basically help to identify all of the neurons that are transmitting odor information from the antennal lobe up into higher brain centers. But in parallel, I was doing something that was a little bit riskier and less well known, which was to study all of the neurons that carry temperature and humidity information. So again, the neurons that sense those cues are located in the antennae, they project into the antennal lobe, and they connect to similar partner neurons that carry them up to higher brain centers, again, used for instinctive reactions or for learning and memory. One of the circuits we found is actually used, we think, for an instinctive withdrawal from a very dry environment, because as you know, insects are very small, they dry out very easily, and that would be quite dangerous for them. Another project I was working on concerned these structures that are involved in learning and memory. These are called the mushroom bodies, and they consist of thousands of parallel cells, and they receive visual inputs, olfactory inputs, the smells, temperature, humidity, and they're used in order to associate these cues with either positive or negative experiences, essentially. More recently, I was leading a project on this structure. So this is the ventral nerve cord. Again, this is the equivalent of our spinal cord. It consists of repeated segments along the body. Most prominently, you can see we have these three labeled bits, T1, T2, and T3. These bumps actually control each of the pairs of legs. And as you know, insects have 6 legs. So I had studied this, the development of this nerve cord when I was a postdoctoral fellow, but this was actually my first opportunity to look at the adult nervous system in detail, and that was really exciting. And I'll just mention one thing that we found. So we know already that there are stem cells that produce neurons that are located repeatedly in these different segments of the nerve cord. And they create these related populations of cells, as you can see here. So these are all the neurons coming from one stem cell in each of these 6 locations. But what we also found was that you can actually identify a lot of individual cells that are repeated between these segments. And I thought these were really beautiful. We didn't have very good, and we still don't have very good ways of identifying them in an automatic way. So this was mainly me looking at all these images and going, I've seen this cell before, and then going and making sure that that was definitely the best match. But then we could also later collaborate or corroborate this by looking at the connectivity patterns between the neurons. Okay, the current project we're working on is an adult male brain and nerve cord. So this is actually, because techniques have become so much faster and cheaper, it's been our first opportunity to reconstruct an intact nervous system. So this is both the brain and the attached nerve cord. So it basically lets us look at all the cells that connect the body and the brain. And we can analyze all their inputs and their outputs at the same time, which was not possible before. It's the first male brain. We had other brains, but those were female connectomes. So now we can identify and analyze neurons that are different between the sexes and that underlie sex-specific behaviors. And one of our focuses right now is the smell and taste circuitry that male flies use in order to assess potential mates. I'm also identifying serially repeating cells and populations that we can find in both the brain and the nerve cord. So we weren't able to do this before either. This is one example shown on the left here. Okay, and finally, I'm looking forward to future work on a different species. Finally, after all of this time, I've worked with Drosphila. So along with, again, Greg Jeffers and also 2 collaborators from the US, I've been awarded a Big Welcome Trust grant in order to study this species, which is Aedes aegypti. It's the mosquito that transmits diseases such as yellow fever, Zika, and dengue. And we're going to be focusing on the neurons that allow them to identify their favorite hosts, which are us, actually. And they do this by sensing body heat, carbon dioxide, and human body odors. So humans are the preferred hosts, and they tend to live in and among humans around the world. Yeah, so I'm very excited to finally have this opportunity to do some comparative work, because although I didn't mention this before, I've actually always been really interested in the evolution of development and basically how different bodies are formed by using the same genes in different ways, essentially. And so this will be my first chance to compare 2 nervous systems that are close enough that we'll be able to find the same cells across those nervous systems, but still be able to study their differences. Okay, so Connectomes required a lot of resources and a lot of collaborators, and none of this work would have been possible without many other individuals and groups and institutions, and these are actually only some of them. I made some changes to the slide, but they didn't make it into this talk, sorry. I'm especially grateful, though, to the Wellcome Trust, because they've funded all of my work in Cambridge, and also in general because they fund these large projects that create resources that are used by scientists all over the world. Anyway, thank you for your attention. I'd be happy to take any questions.